All right, yeah, then thank you very much for attending, and thank you very much to the organizers for this great webinar. There we go. Yeah, I will talk about recent developments in super resolution microscopy, in particular STED, some new inventions from, sorry, just having some. Yeah, about recent developments in super resolution microscopy. So first, I give a speed introduction into STED. As for all fluorescent microscopy technologies, you usually use an excitation laser to switch on the fluorophore, getting it to a higher energy level to get some photons out of it with a specific wavelength. But then, on the other hand, you can also switch off this fluorophore with another wavelength, usually a little bit longer wavelength. And this is basically the principle of STED microscopy. So the clever idea was to introduce a modification like a phase mask and to lay this off switch around your excitation laser in the form of a donut, making sure nothing is excited where the depletion laser is. And then when you look at the 20 nanometer beats using only the excitation laser, you get a resolution of about 221 nanometers. So that's the confocal resolution. If you now power up the depletion laser and lay it around the excitation laser, you basically make the excitation window smaller and smaller and getting down to a resolution to 22 nanometers. And this principle is basically, or all of the products from Aberrio Instruments work with this or a similar principles. So for example, there is the Steadicon, which upgrades your existing wide field system to a confocal microscope, and STED nanoscope with a resolution down to 30 nanometers. And all that is required is basically a free camera port and a good objective lens, as Rimas pointed out in his great talk. That is crucial. It is actually optimal for setting up in your kitchen as well if you want. So if you want to, why not make it a homestead if you buy a confocal microscope. And yeah, we have a demo system in the UK. If this is of interest, please ask me for a remote demo or a live demo if this is possible. That's easy to arrange. Then you have the FacilityLine, which is the most customizable platform. For experienced microscope builders, you can combine STED with other technologies like two-photon, FLIM, optical trapping. The MinFlux is a relatively new technology, was first published in 2017 by Stefan Hell's lab, who is one of the co-founders of Aberrio Instruments, and it can resolve structures down to 2 nanometers in XYZ. And then the FacilityLine, there is the latest innovation is basically set into the FacilityLine, which I will talk later about. So yeah, the Steadicon, I think it's a great microscope. I have one behind me set up. Basically, you have the scanner head that also contains the 405 nanometer laser and the support box, and that's it. And the rest is basically a wide field microscope that can already be in the lab, for example. It is highly compatible with inverted, with upright stands. Nearly all of the stands are possible to upgrade with this STED microscope. And briefly, the specs are, so there are 3 pulse lasers, 4 pulse lasers, 488, 561, 640 as excitation laser, and it has a 775 nanometer STED laser. It's also possible to upgrade to a 405 laser if you want to see DAPI to do 4-color confocal imaging or routine 2-color STED imaging. The 2D STED resolution is about 40 nanometers. The detection, you can have up to 4 detectors. So your imaging speed is quite fast then. And they are using APDs with quantum efficiency about 60% or higher in the red shifted area. You have a motorized pinhole. You can upgrade to AFM or FLIM. And the new options are autofocus, which is great for lifestyle imaging. And I think STED is a great method for doing super resolved lifestyle imaging. Deconvolution, so there's a Huygens package available, and multi-region of interest. So you have a nice big area that you can scan for small regions of interest that you want to super resolve. As I said, the STED detectors are APDs, avalanche photon detectors, which have an outstanding detection efficiency in the redshifted region. And this is why you can basically use a lot of dyes for standard 2-color super resolution imaging, usually excited with 640 and 561. So you basically, for sample preparation, you just use them like a normal secondary antibody. And also for lifestyle imaging, there are a lot of pre-conjugated antibodies where you can see actin or mitochondria, and then the silicon rhodamine, which also works well. So I think, yeah, all in all, it's quite good, good and affordable system to upgrade to confocal and STED resolution. Yeah, some images. So this spectrine rings, which are usually at 150, 120, 150 nanometers apart, which you can't resolve with normal confocal. If you look at them through STED, you can see the rings. And then some mitochondria where you can resolve superstructures, or Bruchpilot, which is a neuronal marker I've worked with a lot during my research in Karlsruhe, Heidelberg, and Bonn, and where you can also see substructures. And then I guess the signature is the nuclear pores, where you can then also resolve to the substructures. Then since April 2019, there is a system called FacilityLine, and this is great, and a great instrument for a multi-user environment with super intuitive software, where even unexperienced users can get a STED image with only a few clicks, but also giving experts full control over all features. And there are some features like adaptive illumination, which helps against bleaching and boosts the resolution even a little bit more. You have 2 depletion lasers, you have 3D STED, and for the face mask, for the bottle beam, they actually use an SLM, a spatial light modulator, which gives you the possibility to correct for aberrations coming from your objective lens in case they are not perfect. And yeah, auto alignment, full push button auto alignment, adaptive optics. I think we will hear later about adaptive optics for deep tissue penetration and brighter signals. And the newest thing since October 2020, so since last year, is the matrix detector, and I will go a little bit into this one. So think of the matrix detector of a detector array of photon counting APDs that measure the signal, basically measures the background and STED signal separately with several different detectors, as opposed to a single point detector that measures background and STED signals simultaneously with the same detector. So this is a detector array, and the problem is always the background. So you have a nice focused and super resolved image in the focal plane, but a blurry background with no useful information from out of focus light at the edges, which gives you a nice artistic effect in photography, but doesn't really help in scientific imaging because it decreases the signal to background ratio. So how it works is, so it gives you access to 2 technologies. One is pixel reassignment, which is the same principle as realized in the area detector. And the second one is differential detection, which also works in STED mode, so in super resolved mode. And the principle is basically regard your 2 eyes as a matrix detector with 2 single units, and then focus on your thumb, close one eye, then close your other eye and open the other eye again. Basically blink between the eyes, and the scene behind your thumb, whatever it is, I hope this is your monitor with this presentation on it, it will jump. And this allows you to distinguish between the foreground, which is your thumb, from the background. And to a single point detector, foreground and background would look the same. And with this method, you basically can subtract then the background, and you will come up with a nice image. So for those of you that can be convinced with pretty images, now a couple of pretty images. So this slide shows different Z slices of a nucleus with stained nuclear core complex with a matrix detector using pixel reassignment, where you still can see a lot of background. And this is with differential detection, where the background greatly disappears. This is Vimentin, also with and without differential detection. And this is the next set of images shows actin in microvilli in confocal mode, STED mode with a small pinhole, and using the matrix detector. And I think this is a great add-on to STED microscopy. Now, the next thing I want to talk about is a technology from a company we work together with. It's the company is called Interherence, and the device is called Vahid. And this is I think a quite nice innovative technology that gives you that tackles a problem that also came up in Rima's talk. So basically, when you want to heat up your cells, when you want to keep them stable at 37 degrees, the objective always acts as a heat sink. And there is a drastic temperature drop in your field of view, specifically in the middle. And the solution is to heat up the objective, but some of the objectives, I think they are quite good for also working at 37 degrees, but some are corrected for room temperature. And the device that they came up for is basically this cover slide. So it's called Smart Substrate. It has the size of a cover slide. It has a heated area in between and a temperature probe. And it only locally heats this small region here, and then it thermally decouples the objective from the sample volume. So you can also have it with a reservoir, or this is a prototype for microfluidics. And it works simply. So you have your microscope adapter here, which has the size of an objective slide. Then your smart substrate with a reservoir, and this is basically your driver connecting it to the controller. It has the footprint of a standard microscope slide. It fits any commercial microscope, so inverted ones. It stays at room temperature, and it electrically connects to the smart substrate. The stability is quite outstanding. If you use an oil immersion objective, then also the heat goes down quite drastically, so you can do very, very precise heat shocks, for example, and you can see over time it stays very precise at 37 degrees. There are some applications for live cell imaging, for example, precise temperature control. This was growing thermophilic organisms, which grow at, I think, I'm not sure, 60 degrees or something. So these are the ones that are also used for PCR, so the enzymes of these ones. And then, of course, you can keep your cells happy for a long time, and as I said, do very, very precise heat shocks, and that's basically it. So with this, I say thank you for listening. And yeah, with the last slide, just want to make all the nice companies visible that we work with and with whose help we try to provide solutions for imaging. Yeah, if you have any questions, please feel free to contact me.